our next speaker is Professor Abhishek Sharma from the Department of Chemistry and Chemical Biology. And the title of his talk is on developing novel therapeutic agents for breast cancer and privileged molecular building blocks. Professor Sharma, you're on.
Thank you, Henry. My talk today is going to be focused on the first project, and so that is our ongoing work in the area of cancer therapeutics. Cancer of the breast is the most common forms of cancer in the female, and more than 70% of these breast cancer are ER positive. So the ER is normally in the off state, and it needs a hormone to go from the off to the on state. And so the chemical structure of the hormone is shown here. This is estradiol, also known as E2. As shown in the structure here, the hormone binds to the ER via forming hydrogen bonds to the polypeptide chain of ER. So shown here is the polypeptide chain of ER, and the hydrogen bonds are also shown more clearly in the lower structure here. You will see that the hormone uses two hydroxy groups in its chemical structure to form these hydrogen bonds. And so this binding of the hormone to ER leads to the change in the conformation and the position of the helix 12 of ER, so as to turn the whole ER structure from the off to the on position. So this is called the on state of ER because ER now becomes capable of starting a cascade of events. So this cascade of steps is shown here. The ER hormone complex forms a dimer, and then this dimer translocates to the nucleus. Here it forms a complex to the coactivator proteins to turn into a transcription complex. This transcription complex turns on the genes for cell cycle. Now, if the concentration of this hormone E2 and the ER complex is too high, then it leads to too much stimulation, overstimulation, and this leads to breast cancer. Some of the compounds used in hormone therapies for ER-positive breast cancer are shown here. However, a few years after the hormone therapy, cancer often comes back in the form of the metastatic breast cancer. This cancer is very hard to treat, and it is fatal. The main reason why these cancers are so hard to treat is that the ER in these cancers has changed. There are some changes in the polypeptide chains of ER. These changes or the point mutations in the ER structure are shown here. And so what these changes does is that these changes force the ER to convert from the off to the on state on its own. And so this means that there is no need of the hormone now. So the ER can automatically convert from the off to the on state and be in that state all the time, constitutively. To solve these challenges, my lab's work has been focused on the development of new classes of compounds for stopping the action of these mutant ERs in breast cancer. The hypothesis behind our first series of compounds was to use this carbocyclic core. This carbocyclic core has a phenol and a side chain scaffold. The phenol motif of this core forms crucial hydrogen bonds with ER. We also use the side chain on this core scaffold to change the conformation of ER and to lead to the degradation of ER. Shown here are three representative structures from our first series of compounds. Also shown here are the IC50 values, anti-proliferation assays in MCF7 cells having wild type ER or the Y537S mutant ER. So the lower the number of these IC50 value, higher is the potency of the compound. So this data shows that seemingly small changes in the chemical structure of a molecule can lead to huge variations in the potency of the compound. So we found that the potency of this compound is lost if you change this polar group. This is the sulfoxide group. So if you change that sulfoxide group with a carbon group here, the potency is totally lost. Also, changing the size of the linker length from the carbocyclic core to this polar group. So shown here, this is a 4-carbon linker. So if you change that 4-carbon linker to a 3-carbon linker, so this also lowers the potency as shown by the IC50. So we have an ongoing collaboration on this project with Dr. Chandra Lapati at Sloan Kettering Hospital. For gaining a better understanding as to how these molecules bind or how these molecules work on ER, we do molecular modeling. Shown here are the two models of the two compounds I showed in my earlier slide. And so this molecule, as I showed, was more potent. So this compound has a C4 linker, and the other compound has only a C3 linker, which means there are only 3 carbon atoms from this oxygen to the sulfur. So these models show that the compound with the C4 linker forms a key hydrogen bond with the tyrosine of ER. And that hydrogen bond is not possible in this compound, which has a C3 linker, because the polar group here is so far away. So we use all the lessons learned from the first series of compounds to further optimize the potency of these compounds. As you can see, the compound, or the best compound from the first series, was potent on the wild type, but not that effective on the mutant ER. The IC50 on the mutant ERs was very high. For this whole process, we use a workflow. We design and synthesize these novel compounds, followed by testing of these compounds, and then followed by the molecular modeling. Shown here is the data from 2 more series of compounds. So we found that changing the structure of the side chain from the linear chain to a cyclohexyl system, shown here, leads to a boost in potency, as shown in the IC50 values. We used this promising data to make the compounds in the series 3 using the same workflow of synthesis, testing, and then molecular modeling. And so this whole process led to a highly potent compound codenamed S31. Comparing the IC50 values in the proliferation assay of this compound S31, you can see that this compound is more than 100-fold more potent as compared to the compound in series 1. If you compare the wild type IC50. And also more importantly, this compound S31 is also very potent on the Y537S mutant ER. And so based on the lessons learned from the compounds in the 3rd series, we are now trying to further optimize the potency and the structure of the compounds, followed by their testing in cell-based and in vivo assays. Finally, I would like to thank all the past and current members of my lab. This work would not have been possible without their commitment and hard work. This completes my talk, and I will be happy to take any questions.
And I don't see any questions posted. And so how far are you from clinical trials involving human subjects?
So Henry, so that will take, I think, at least 2 years. Because if I go back to that structure, we have the structure S31. We further need to optimize this particular compound and want to see how they behave in terms of the metabolic assays. And then there's a whole suite of other assays which we need to screen. So it will take a few more series of the compound so as to, so as to reach to the phase 1 trial stage.
Okay, well, I certainly wish you the best of success. We do need clearly highly effective drugs, right, for treatment of breast cancer. Thanks for your great work.
Thank you.